Recently, science has been revealing remarkable new insights into the human body. There is a vast web of information networks, just like the internet, within our bodies. Where our various internal organs are communicating directly with one another through the exchanging of messages. It seems that these communications are also deeply involved in the processes that take place in the very early stages of our lives. How exactly does such a complex, elaborate system develop from just one tiny fertilized egg? This may be one of the biggest questions that scientists the world over have been trying to answer. Cutting-edge research has been shedding light on some incredible mechanisms buried deep within the fertilized egg. And the key players are the signaling molecules that are being used by our cells to communicate with one another. It's now understood that key messaging molecules are released at different stages so that our organs can start taking shape on their own.
I think the surprise for me was the amazing degree to which the cells sort of know what to do. They're constantly communicating with one another and instructing them what to become.
Moreover, these exchanges can have a substantial impact on the mother's womb, too.
So part of the placental cells seek out the mother's blood vessels and invade the walls.
In the struggle to survive, the unborn baby reacts in astonishing ways. The veil is finally being lifted on what had been completely unknown before. The incredible dramas of life before birth. Join us to witness this incredible story. December 2016. We are going to follow a couple having a child from the earliest stage of the pregnancy. The Matsunagas, Atsushi and Megumi. They were married 3 years ago. As Megumi had to have surgery for uterine fibroids in the past, they had been having trouble conceiving. And a gynecological exam revealed further obstacles.
I was told that my egg cell count was very low. They recommended we conceive within 3 years.
In order to make the most of a narrow window for conceiving, the Matsunagas made a decision.
We're going to try IVF.
IVF is for in vitro fertilization. It means Megumi's egg is fertilized with Atsushi's sperm in a petri dish. This is a clip of that very moment. On the bottom left of the screen is a sperm cell. It's advancing towards the egg cell. It makes contact. But this is just the beginning. Two round objects have appeared. These are cell nuclei, each containing the genes of the mother and father. The genes that were packed into the head of the sperm cell balloon outward. And then the contents of the two nuclei suddenly vanish. It is at this very moment that a new life begins. This miraculous moment was captured using a special microscopy technique. Let's take a look at this breakthrough footage, the first of its kind to be broadcast. This is the very moment the two nuclei vanish. The individual chromosomes containing genetic information are clearly visible. They're now gathering together, and the genes of the mother and father merge to create a new life. 2 hours later, the egg begins the cell division process. Over 5 days, the number of cells grows to around 100. After this, it's time for implantation. The fertilized egg has been returned to Megumi Matsunaga's womb.
最初これを渡されて、今度受精卵を戻しましたっていうので。
Now comes the most challenging part of the process. Will the fertilized egg implant itself properly in the uterus wall? Fertilized naturally or in vitro, an egg must implant itself for the pregnancy to be successful. In clearing this difficult stage, a signaling molecule comes into play in the womb, and the change it triggers is dramatic. So what happens here? This decisive moment was captured by this scientist, and it was a world first. Dr. Ali Brivenloo specializes in the study of the fertilized human egg.
We had never seen this before because, again, it's impossible to have access to this biological material, especially knowing that women don't know even that they're pregnant.
Dr. Brivenloo creates an environment like the uterus in a petri dish. Then he observes the fertilized egg after implantation. A substance, shown here in yellow, begins to pour out from around it. Over a period of days, it keeps increasing. This is a molecule called HCG. The egg uses it to send the mother's body its first messages, saying, "I'm here." This message plays a crucial role in the egg taking root in the uterus. The egg implants itself to the wall of the uterus. But without intervention, the next menstrual cycle would flush it out of the body. So the egg releases a signaling molecule, HCG. It enters the mother's bloodstream and acts on the ovaries and other organs. This blocks the next menstrual cycle and instead causes the wall of the uterus to thicken. The egg then becomes firmly entrenched. The baby egg calls out, and the mother's body answers it. That's what the implantation process is.
These cells are those who are going to be talking to the maternal cells to establish all the inductive interactions and communications that are required.
It's been 3 weeks since the fertilized egg was returned to Megumi Matsunaga's womb. She visits the doctors to find out whether it has taken root properly in her womb. Only 1 in 3 IVF treatments is successful on the first try. Here are the results of her blood test. They show a large amount of the molecule HCG, the one that the baby uses to signal, "I'm here." The fertilized egg cell has been firmly established in the soft tissue of the thickened uterine wall. Inside Megumi Matsunaga's womb, a fetus has begun its journey. And this is only just the beginning. There's still a lot of work to be done. Our bodies are made up from many different kinds of cells. They come in all shapes and sizes. Stomach cells, gut cells. We live and breathe because of these cells working away inside our organs. But all these different cells have actually come from the division of just one fertilized egg. So how exactly was such a thing possible? We now have the answer. Quietly behind the scenes, our cells are sending messages to each other. This is Kenneth Chen, a world-leading expert in the field of regenerative medicine. For the research into the beginnings of life, specific types of cells are now available.
The major advance, of course, was the discovery not only of human embryonic stem cells, but human iPS cells. Of course, Dr. Yamanaka in Japan was the pioneer in this area, and this has had a transformative effect in our field.
Previously, researchers in this field could only use embryonic stem cells, or ES cells in short. ES cells are based on cells that have been extracted from a fertilized egg. And that is why it has the natural power to transform itself into any cell. Dr. Shinya Yamanaka in Japan was the first scientist who had succeeded in developing induced pluripotent stem cells, or iPS cells in short, by adding special genes to common types of cells, like adult skin cells, and growing them in a cell culture. The cells with pluripotency can be produced. They are able to transform into any cell in the human body, just like ES cells. By using these 2 types of stem cells, that is ES cells and iPS cells, researchers are now able to observe the process of how organs are formed from a single cell in a petri dish. At Chen's lab, ES cells are used to observe the formation of one organ almost every day. Here, the researcher adds a certain signaling molecule to an ES cell. It's called WNT. WNT molecules are present in large quantities in dividing eggs. After 1 week, part of the cell is starting to pulse. Two days later,
2つなんですけどね、拍動しているのは肉眼でもわかるぐらい。
large ripples move across the flattened cell. This is the very beginning of a human heart.
You can take an embryonic stem cell that can become any cell and then instruct it to specifically, almost entirely become human beating ventricular muscle. This is an exciting event. And those instructions generally are going to be, I believe, signals that come and tell the cell to go in one direction or another.
The first organ to form is the heart. What do you think comes next? A possible answer to that question presented itself right in front of our cameras. Dr. Takanori Takibi is a researcher who is making waves in the scientific world. He uses iPS cells. He has successfully induced them to turn into cells of some different types of organs. Today, he is observing heart cells made from human iPS cells.
面白い。これですね。この拍動してない子たちはもしかしたら肝臓のこう初期的な構造物が出来上がりつつあるのかもしれない。
Right next to the pulsing heart tissue, something quite different has appeared. Further examination confirms that it's made up of liver cells. The heart cells stimulate neighboring cells to become liver cells. The heart cells are sending out signaling molecules with the message, "Become the liver." And when these messages are picked up by the neighboring cells, they form into the liver. It's becoming clearer that as these molecules are exchanged back and forth in an increasingly complex manner, organs like the lungs and pancreas are then created, one after the other.
The human body is never building just one organ. Various organs, whether close to each other or far apart, are all communicating.
But questions still remain, such as, after a certain type of cells are produced, how do they grow into an organ with a very complex internal structure? These are the laboratories of a globally known German medical technology company.
Oh, hello, welcome.
Dr. Klaus Engel is a specialist in visualizing the world inside the human body. These intriguing images show the body as if we're looking straight through it. Enormous amounts of imaging data from a patient's entire body is used. It's hard to identify which part of the body each image comes from to untrained eyes. Dr. Engel combines these images in 3D and uses a special technique to add color. Once this is done, photorealistic images come to life. These remarkable visuals have been created from real medical data from the human body. This is the heart. And this here shows large blood vessels inside the brain. Let's take a look inside an organ. This is inside a lung. The human bronchial tubes are very complex structures. They split again and again, over 20 times as they branch out into smaller, thinner airways. So how do such internally intricate organs get created? There is a researcher in Japan who has been trying to get to grips with this mystery. This is Dr. Takashi Miura of Kyushu University. He has been exploring how bronchial tubes develop using computer simulations. Previously, bronchial tubes were thought to start branching out just by receiving a positive instruction, stretch, from neighboring cells. However, he found that just with this message to urge tracts to stretch out, it's quite hard to complete the process of creating the whole branching system. To make the branching effect take place, he needed to build up an extremely complicated computer program. So Dr. Miura decided to try something else. Along with the message, stretch out, he introduced another message in his simulations and saw what would happen. This second message was a negative one. Won't stretch. When he ran the simulation again using these contradictory messages to make counter-regulatory actions happen, he discovered that even a very simple program was able to initiate the branching effect and successfully recreate the structure of the bronchial tubes.
As signals are exchanged, rules are made about shapes, and these branches form by themselves. Even a very simple, two-way interaction can result in a structure like this.
It's now been 2 months into Megumi Matsunaga's pregnancy. Within Megumi's womb, too, this communication is taking place. At this point, the baby is only 3 millimeters in size. But already, only a few weeks into pregnancy, the first organ to form, the heart, begins to beat. This is the starting point of the domino effect, forming the full lineup of organs. Signaling molecules are flying everywhere within the developing body of the baby. We can now see the arms and legs. Once again, this is the work of signaling molecules. Meanwhile, what's happening inside the baby's body? Fast forward another 2 months. And you can see other organs at the area around the heart. The liver is directly below it. The lungs and intestines have also been formed. The stomach appears in black, being filled with amniotic fluid. Research has revealed that an assortment of different signaling molecules are released, one after the other, causing cascading reactions and leading to the formation of our organs by themselves. The timings and the amount our cells release such molecules seem to be very precisely adjusted. In the womb of the mother, an extraordinary mechanism is unfolding. This knowledge gives us an entirely new perspective into the story of our life before birth. The baby growing inside Megumi's womb is only just halfway through its journey. since starting out as a fertilized egg. A big task waits for both of them around the corner.
最初のこの辺は本当に虫みたいだよね。この辺からちょっとずつ宇宙人になってきたかな。よいしょ。
And the next task is growth in size. If a fertilized egg were the same size as an apple, about 10 centimeters across, how big would a newborn baby be? The baby would be 500 meters tall, higher than the Empire State Building. A baby's growth over 9 months is quite extraordinary. This rapid growth is supported by something crucial. That's the placenta. The area where the tip of the baby's umbilical cord connects with the mother's womb. The placenta is part of the baby's body, so it also has developed from that single fertilized egg cell. It's an organ where the baby receives oxygen and nutrients essential to its growth. Let's take a look to see what's happening inside. Inside the placenta are tree-like structures. We'll call them the baby's branches. The baby's blood vessels run through these structures. This is the frontier of the baby's own body. Looking above is the wall of the mother's womb. The holes that can be seen are openings that connect to blood vessels of the mother's body. We'll call them windows of riches. The baby's branches absorb nutrients from blood flowing from the mother. Oxygen is also absorbed indirectly through the branches. When red blood cells collide with them, the branches strip away the oxygen the cells are carrying. The mother also sends out messages. Our old friends, the signaling molecules. In response to the mother's stretch out messages, the baby's branches are encouraged to spread out. More oxygen and nutrients then become available, allowing the baby's continued growth. Cutting-edge research has been revealing more and more of these signaling molecules, which are at work inside the placenta. Detailed genetic analysis are now helping us create a complete picture of molecules being exchanged in the placenta.
So what we see is interesting is a lot of crosstalk going from both sides. So the maternal and fetal side communicate a lot from the mom to the fetus, as well as, of course, a lot of signals also coming back on the other side, from the fetus to the maternal side.
Staggeringly, over 100 signaling molecules have been discovered so far. Scientists have been trying to decipher their functions. And they discovered that one of them has the power to dramatically sway the baby's growth. This molecule causes startling changes inside the mother's womb. Megumi Matsunaga is entering her 4th month of pregnancy.
How are you feeling? The morning sickness is pretty bad. I'm throwing up anything I eat and I can't keep fluids down.
It will be about a month until her condition stabilizes. During this period, every mother would be wondering if her baby is growing and doing well. Now the baby is about 10 centimeters long. And inside the baby's head,
ここまで大脳なんです、この薄いのは。ちょっと厚くなってきてるのが分かりますかね。
the cerebrum is the part that looks like a thin casing around the brain. There's a big gap between it and the core of the brain. There is still much growth ahead for the brain and other organs. At this stage, something astonishing is happening inside the womb. To carry the baby, the mother's body literally bears pain. And the pain comes from the baby's branches. Dr. Susan Fisher is the one who shed light on this process. Her research focused on those windows of riches. The openings where blood vessels form an outlet into the womb. These are electron and microscope images of windows of riches. On the left is early stage pregnancy. On the right is from the mid stage. It may not look much different, but when we show them at the same scale of magnification, the difference becomes clear. The opening on the right is 10 times bigger. Further investigations revealed a remarkable phenomenon. As the window of riches expands, cells, slightly different from the surrounding ones, start appearing. They are shown here in yellow. Let's take a further look in close-up. These cells, in fact, come from the baby. These type of cells are normally found in the baby's branches, but they are now gathering in the wall of the mother's womb in large numbers.
So part of the placental cells have specialized molecular homing devices to be able to seek out the mother's blood vessels and invade the walls.
In the mother's womb, such a dramatic event takes place, which will then spur the growth of the baby. Let's take a look at that pivotal moment. We are inside the placenta of the fetus in its 4th month. The baby's branches have grown so large, they are now more like baby's trees. They have spread out an impressive crown of branches. However, the branches have turned slightly black. As the baby grows larger, the mother's blood supply can no longer deliver enough nutrients and oxygen. Without an adequate supply, the baby will no longer be able to grow. There is a severe bottleneck. But then something starts being released by the branches. It's a signaling molecule called PGF. It sends the message, I need to grow bigger. The message is received by the mother's uterus. Then, in response, windows of riches open wider, allowing a larger volume of blood to flow. But this big event isn't over yet. The tips of the branches reach out even further, right to the uterine wall. Cells from the baby's branches penetrate the wall of the uterus. And then reappears on the inside of the window of riches. The cells shoulder their way through the walls of the window. And then the walls of the blood vessel crumble to create a larger opening for the parental pipeline. Now, huge amounts of blood wash over the baby's branches. This is such an aggressive event, but necessary to release a life-giving shower. Part of the mother's body is literally torn open. The baby's branches can now absorb plenty of oxygen and nutrients. They pass it through the umbilical cord to the fetus. And this enables the fetus to grow much larger. This bleeding change should also be happening inside Megumi's body to protect her baby. But Megumi has been worrying about something. It's to do with surgery that she had before she became pregnant.
There were over 10 uterine fibroids. So there was even talk about a hysterectomy. So when I was told that I might not be able to have children, that was so tough.
Megumi managed to avoid hysterectomy, but the surgery to remove the fibroids resulted in a much thinner uterine wall. So she had been worried whether her womb was able to keep supplying large amounts of blood that the baby needed to grow. They're going to check the blood flow to the placenta.
これ胎盤ですよね。血液がしっかり流れ、酸素と栄養が運ばれると。
Megumi's womb had been supplying large volumes of blood to the baby. As a result, the baby has grown considerably.
脳がいっぱい詰まってますよね。この辺全部大脳ですよ。
An area that was an empty space at the last scan is now filled with brain cells.
もういつ生まれても大丈夫なように準備ができてる。
The due date is now just 1 month away. The baby's brain is growing and developing inside the mother's body. When you compare the size of a baby's brain with an animal who shares our ancestry, like a newborn chimpanzee, the human baby's brain is 3 times larger. Our large brains have always been the driving force behind us, helping us build advanced societies and prosper so much. And what have helped our brains' highly sophisticated workings are those signaling molecules. In our brains, too, they are acting in a complex and amazingly elaborate way. The effect is no less remarkable than the miracles of the beginning of life. Now, another stunning world is about to be revealed. To make the brain's works visible, Naoki Matayoshi, a well-known Japanese comedian and writer, was invited to take part in an experiment. Naoki's sketches and books have been hugely popular in his country. Full electrical impulses crisscross the brain along these neural pathways. How exactly does the brain use this intricate network? Here's an example. When Naoki sees the face of someone he knows on the street, acid, and it's also a signaling molecule. And the message they carry is fire with new ideas. Messaging molecules are key to our cutting-edge understanding of the complex and sophisticated workings of the human brain, too. It's Megumi's delivery day. As Megumi had surgery for uterine fibroids before her pregnancy, her baby will be delivered by cesarean section in order to minimize any damage to her womb. An hour into the delivery, the newborn's cries ring throughout the delivery room. It's a very healthy girl. The baby that had started out its life as a tiny fertilized egg and cared for preciously in her mother's womb. It's nice to meet you, finally. The beginning of life is full of wonder and awe. The new knowledge about signaling molecules involving fetus development has also led to some revolutionary treatments to save the lives of infants. Landry Mulholland is 12 years old.
Hi, Landry. Can I introduce you to a friend?
She suffers from an impaired function of her bladder, intestines, gallbladder, and other organs.
She has a hard time being able to eat because the nerves in her bowel, they don't squeeze.
Doctors think it may have been due to disruptions in some signaling molecules before she was born.
Right here, you get an IV every day where you get nutrition. Can you show them? So what's it like to get TPN?
Well, it's kind of weird, but it helps. So.
How long are you on TPN a day?
Landry has had numerous operations, but recently, the condition of her liver started to worsen.
Liver tissues in culture.
Dr. Takanori Takibi is now involved in her treatment. As we saw earlier, he conducts research on the process of organ development.
We can save you by your own liver tissues you can growing.
Yeah, watching a baby grow and develop and it's cool.
Cool.
Isn't that cool?
Yeah, that's kind of what it's like.
Thank you so much, Landry. Thank you so much. Nice meeting you.
Thank you
it's
for
my great
everything.
pleasure.
Yours too. Mine too.
Right. Take care.
You too. Bye.
Good day.
Dr. Takibi is developing a treatment that uses iPS cells to create a mini liver. The idea is that when it's placed in the patient's ailing liver, it will begin to help regenerate itself. This is at the frontier of regenerative medicine, being tackled by researchers around the world. But just making her liver cells is not enough. He has to make liver tissue that functions as an organ. The liver is a very complex organ. This footage of living liver cells was taken using an 8K microscope. Visible in blue are liver cells. The other cells around them in red are capillaries. For the liver to function, it needs this complex tissue structure. But the structure is just too challenging to be created artificially. So Dr. Takibi's method is to simulate what happens in the mother's uterus. He uses iPS cells that are in the process of changing into liver cells. They are mixed with blood vessel cells and supporting tissue cells. This creates conditions similar to those when the liver forms in the fetus. Then he cultures the cell mixture in an environment that is similar to the mother's womb. Dr. Takibi hopes that this will induce the cells to send out signaling molecules and trigger the process of building the liver's complex structure. After many experiments, the dispersed cells begin clumping together on their own.
きれいきれい。やっぱり締まってますよ血管。そうですね。違いが出てる。出てる出てる。
The product is a tiny liver, just a few millimeters in size, with a network of blood vessels inside. But will it function inside the living body? The tiny livers were implanted in mice with impaired liver function. This operation dramatically increased their 30-day survival rate. A miniature liver that functions in an animal has been successfully created. This is a world first. In 2017, for the next step toward treating people, Dr. Takibi launched the lab that can mass produce these mini livers. He hopes to start human trials in 2020 on children suffering from serious liver diseases.
The purpose of our research is to save the lives of children like Landry. I hope we will find a cure for them using these mini livers. I expect that this method will become available for clinical use in the very near future.
Researchers around the globe are working rapidly to create these artificial organs called organoids.
So these are stomach organoids, and these are intestinal organoids.
Here is a stomach organoid that releases stomach acids. And this intestinal organoid is showing an involuntary muscular wave-like movement needed to pass food through the gut. Even the kidney, known to be one of the most complex organs in our body. A part of its structure has now been successfully created in a laboratory.
These organoid technology gives us a window to understand each of these different organs, and ultimately it will be important to synthesize all of this together to get a more complete picture of the mysteries of the human body.
Precious, irreplaceable lives continue to join us every day. Each one of us is the product of so many miracles. You also started your journey from just one fertilized egg. This is where your story began. One by one, as signaling molecules were released, your various organs started forming. And when you sent out messages to allow you further growth in the womb, your mother's body responded, continuing to supply you with her riches. You are the result of a highly complex, sophisticated workings of the human body. Megumi Matsunaga's baby is now 5 months old. She is now 3 times her birth weight and is getting bigger. A remarkable exchange of microscopic messages takes place between our internal organs to sustain our lives in an unimaginably elaborate and complex manner. As we gain further insights into our body's inner networks, somehow, life feels more precious than ever.